So today we will be going over enzyme inhibition, which is a part of the HL biology syllabus. Firstly, inhibition is the preventing or slowing down of enzyme-catalyzed processes, and so this can actually be quite helpful in many ways. When we look at medicine, it can be used for treatments and the development of new drugs. This is because inhibition allows us to look at specific enzymes that can cause processes associated with disease and then design drugs or treatments that can inhibit these specific processes. Inhibition is commonly divided into two main subtypes, which are competitive and non-competitive. Looking at competitive inhibition, the key point associated with it is that the inhibitor competes with the substrate for the active site of an enzyme. And what this means is that the inhibitor is therefore chemically similar to the substrate, so that it can bind to the complementary active site. Essentially, the competitive inhibitor can bind to the active site of an enzyme and then physically block the substrate from binding to the same molecule, and this prevents the process from occurring as fast as usual. This is especially impactful when substrate concentration is lower, however less so when it increases, as I will describe later. One example of a competitive inhibitor is the use of statin drugs to lower cholesterol levels. Statin is a competitive inhibitor for the HMG-CoA reductase enzyme, which helps synthesize cholesterol. And so when it binds to the active site of the enzyme, it therefore prevents the substrate from binding and reduces the production of cholesterol. The second broad type of inhibition is non-competitive inhibition. And different to competitive, the inhibitor in this case now binds to an allosteric site, as shown in the diagram. And this is a site that is separate from the active site. Hence, in this case, the inhibitor is not required to be chemically similar to the substrate. In general, it is important to note these differences, as compare and contrast questions can ask about the differences between the two. Now, when a non-competitive inhibitor binds to the allosteric site, there is a conformational change in the active site, which makes it no longer complementary to the substrate. And this is seen in the diagram, where the red substrate can no longer fit into the enzyme. This therefore limits the rate of reaction. Going forward into more comparisons between the two, on this page we have a graph showing the rates of reaction for uninhibited, competitively inhibited, and non-competitively inhibited reactions. The y-axis shows the rate of reaction and the x-axis shows the substrate concentration. So we see that for competitive inhibition, as substrate concentration increases, the maximum rate of reaction can eventually become the same as it is without the inhibitor. And this is because there will eventually be more available substrate than inhibitor. However, a key difference is that in non-competitive inhibition, the reaction rate remains low even as substrate level increases. And this is because the altering of the active site caused by the binding of the non-competitive inhibitor to the allosteric site simply leaves fewer enzyme molecules available to catalyze reaction. So even if you have more substrate available, there will only be a limited number of enzyme molecules that will be able to bind to them. Now we will be looking at two more cases, which are end product inhibition and mechanism-based inhibition. So for end product inhibition, the key concept is that the end product acts as a non-competitive inhibitor. So in these processes, it is used to ensure that cells manage their resources well. And here, when the end product acts as a non-competitive inhibitor at the first stage of production, it can prevent an overproduction of certain molecules. The example shown on the left is of the case of isoleucine, which is an essential amino acid. And in this case, when enough of it is produced, the isoleucine molecules can non-competitively inhibit the enzyme that produces them, and this therefore helps regulate the amount produced. Now, the second type of inhibition is mechanism-based inhibition. In this case, the inhibitor binds irreversibly to the active site through a covalent bond, and this produces a complex which is stable and can permanently remove an enzyme's catalytic ability. So this form of inhibition is actually irreversible, and that is a key difference compared to, for example, non-competitive inhibition. Finally, we will wrap up with a key example for mechanism-based inhibition, which is penicillin. So penicillin irreversibly binds to transpeptidase, an enzyme in bacteria that helps maintain the rigidity of the cell wall. When penicillin binds to it, as a result, the stable complex that is formed removes the catalytic ability of transpeptidase, and therefore the cell wall rigidity of bacterial cells are no longer maintained, and bacterial cell walls burst, leading to bacteria dying. And so through this, we can once again see how the concept of inhibition can be used to develop treatments.